All right, welcome. Today, we're going to open up the molecular case file on SARS-CoV-2. We're going to deconstruct this virus piece by piece, all the way from its family origins to its really remarkable evolutionary trajectory.
So here's our game plan. We'll start with a viral family portrait to get our bearings. Then we'll move on to some genetic fingerprinting to trace its lineage. After that, the really cool part, dissecting its art of invasion at the molecular level. Then we'll look at its replication masterclass, and we'll wrap up by tracking how it continues to evolve.
Right. First things first. To really get a handle on our subject, we need to meet the family. Where does SARS-CoV-2 actually sit within the vast, and I mean vast, world of coronaviruses?
And vast is a serious understatement. The coronavirus family is immense. We're talking estimates of over 5,000 distinct viruses. And let's be honest, that's probably just scratching the surface. To give you some perspective, over 500 have been found in bats alone, which just highlights the enormous viral reservoir that's out there in nature.
Now, what's really interesting is how these thousands of viruses are grouped. We've got 4 main genera: alpha and beta, which mostly infect mammals, and then gamma and delta, which you'll typically find in birds. Our subject, SARS-CoV-2, belongs to the beta coronaviruses, a group that, as you can see, is pretty notorious for containing some serious human pathogens, like the original SARS and MERS. Okay, let's turn to a bit of genetic forensics. We're going to use the virus's own genetic code, its RNA, to build its family tree and trace its lineage with molecular precision. A phylogenetic tree is, for all intents and purposes, a molecular fingerprint. By lining up and comparing the RNA sequences of different viruses, we can measure how far apart they are evolutionarily. The shorter the branches between two viruses, the more recent their common ancestor. This is an absolutely fundamental tool in virology for tracking down where these things come from. And what these sequences reveal is just incredible. We can establish shared ancestry, we can calculate the virus's mutation rate to create what we call a molecular clock, we can reconstruct transmission chains in an outbreak, and, critically, we can identify those zoonotic spillover events. That's the moment a virus jumps from an animal to a human host.
And this tree illustrates that point perfectly. It maps out the human coronavirus family. You can clearly see the seasonal, milder viruses like OC43. But then, you have the more severe beta coronaviruses. Notice how SARS-CoV-2 and the original SARS branch from lineages with incredibly close relatives found in bats. This is the crucial genetic evidence, the smoking gun, that points right to their likely origin. Now for the main event, the molecular break-in. How exactly does SARS-CoV-2 use its sophisticated protein machinery to gain entry into one of our cells?
The virus's number one tool is its spike protein. But don't think of it as a static key, it's more of a dynamic, shape-shifting machine. As you can see, it flicks between two states, a closed or RBD down conformation, where it's pretty stable and shielded, and an open or RBD up state. It has to adopt this open state to expose its receptor binding domain and actually kick off the infection.
This whole invasion follows a precise 3-step sequence. First up is binding, where that open spike latches onto our ACE2 receptor. Second is cleavage, where our own enzymes cut the spike protein, unleashing its fusion machinery. And finally, fusion, which is this dramatic structural refolding that merges the two membranes. Let's watch this unfold. Here, in step 1, we see that initial critical contact. The spike's receptor binding domain, now in that up position, has found its target on the cell surface, the ACE2 receptor. The key is in the lock. That binding event is the trigger. It kicks off an irreversible and incredibly dramatic change in the protein's conformation. It sheds part of its structure and begins to refold into a completely new shape, getting ready to execute the final part of the invasion. And here is the end game. The refolded spike proteins now act like little molecular grappling hooks. They harpoon the host cell membrane and physically pull it towards the virus. The two membranes fuse, creating a channel for the viral RNA to be injected directly into the cytoplasm. Invasion complete. So what makes SARS-CoV-2 so good at this? Well, it has a key feature not found in its closest bat virus relatives: a furin cleavage site. Furin is one of our own enzymes, and it's abundant in our respiratory tract. This site allows the spike to be pre-cleaved as new virions are being made, basically priming them for action before they even leave the cell.
This makes that whole fusion process incredibly fast and efficient when they find their next target.
Once it's inside, the viral genome doesn't waste a second. It executes its master plan, hijacking the cell's own machinery to turn it into a dedicated factory for producing thousands and thousands of new viral copies.
This slide lays out the entire factory floor plan, so let's walk through it. Once the viral RNA is released, it's immediately grabbed by the host's ribosomes to make the viral polymerase. This polymerase then sets up shop, creating a full-length negative strand copy to replicate the genome, and also transcribing a whole set of smaller messenger RNAs for the structural proteins, like spike, membrane, and envelope. These proteins are trafficked through the cell's own systems, where they assemble with the newly copied genomes. Finally, new, complete virions bud off, ready to infect more cells. It's a true masterclass in biological efficiency.
All right, finally, let's look at the surveillance operation. How do we detect this virus, and how do we keep track of it as it constantly changes its disguise to evade our immune system?
We have two main diagnostic tools. First, the gold standard, RT-qPCR. This is a technique that detects viral RNA with extremely high sensitivity because it amplifies the genetic material. Second is the rapid antigen test. Instead of RNA, it detects viral proteins, usually the nucleocapsid protein, because there's a lot of it. It's much faster, but since it doesn't amplify anything, it's just inherently less sensitive.
And you can see the difference in complexity right here. On the left, the whole lab process for RT-qCPR, sample collection, RNA extraction, amplification. On the right, the elegant simplicity of a lateral flow device. Both are absolutely essential. They just play different but complementary roles in public health surveillance.
But tracking isn't just about presence or absence. It's about evolution, this constant pressure from our own immune systems, whether from getting sick or from a vaccine, is the main thing driving the virus to change. The virus is forced to change its disguise to survive.
And this just brilliantly illustrates that evolution in action. You can literally see the spike protein accumulating mutations. Compare the original wild type on the left to Delta in the middle, and then look at the explosion of mutations in Omicron on the right, especially on the receptor binding domain. Every single one of those changes is a potential escape from our antibodies. Which brings us to the final and most critical question. We've deconstructed this one case file, but the wider coronavirus family is still out there in its natural reservoirs, constantly producing these little sparks in the form of spillovers. The grand challenge for virology and public health is to learn how to predict which of those thousands of sparks has the right molecular machinery to ignite the next global fire.